By combining in situ hybridization and tetramer staining in a technique known as ISTH, the relative numbers and spatial relationships between viral antigen-specific immune effector cells and virally infected cells and tissues can be determined. ISTH begins by embedding fresh SIV-infected tissue in agarose. The tissue is cut into 200 micron thick sections using a vibratome and stained for viral antigen-specific CD8 positive cells using FITC-labeled MHC tetramers. After freezing, these stained sections are further cut into 8 micrometer thick sections using a cryostat, and in situ hybridization is performed to localize virally infected cells. Confocal microscopy is used to image the double-stained sections and ultimately reconstruct the original thick sections to reveal the relationship between immune effector and target cells. Using this procedure for examining early SIV infection has proved to be a good predictor for the severity of the ensuing disease.
Hi, I'm Qingsheng from the laboratory of Ashley Haase in the Department of Microbiology at the University of Minnesota.
I'm Pam Skinner, an assistant professor in the Department of Veterinary and Biomedical Sciences at the University of Minnesota.
Today, we will show you a procedure for in situ tetramer staining and the in situ hybridization, or ISTH for short.
In addition to using in situ tetramer staining combined with in situ hybridization, as we're demonstrating today, we also routinely use in situ tetramer staining combined with immunohistochemistry to determine the location, abundance, and protein expression profile of antigen-specific CD8 T cells in situ.